Hey guys, so this lab is about gram-positive diagnostic tests. The reason we are learning is, you have a patient with a bacterial infection, you did the streaking, you separated the bacteria as single colonies, you did the gram staining, and when you saw a lot of gram-positive cells, your next step is to identify the bacteria. That's the reason the lab 3 is all about gram positive diagnostic tests. Okay, so you did the streaking, you did the gram staining, and then you're doing some gram positive diagnostic tests. So today's lab is all about gram positive diagnostic tests, and later on we'll talk about gram negative diagnostic tests. So let's begin with some of the very commonly done gram positive diagnostic tests, and the first one is the MSA test, mannitol salt agar test. Before we know anything about this test, let's quickly take a look at the different plates that we have. So far, you are familiar with the TSA media, tryptic soy agar media. Tryptic soy agar bacterial media is an example of all-purpose media. By all-purpose, what I mean is any bacteria can grow on this plate because this is a very rich, nutrient-rich media, and any bacteria, E. coli or Staph aureus or enteric bacteria or skin bacteria, aerobic bacteria, any kind of facultative aerobic bacteria can grow on TSA plates. Okay, it's all-purpose media. But in order to identify the specific microbe, we need to modify the media so that it can select for a specific microbe and restrict the growth of other microbes. One such kind of bacterial media is known as the selective media, and the very good example of the selective media is mannitol salt agar media. Okay, this is not only selective, but it is also differential media. So let's take a quick look at what is the selective versus differential bacterial media. Selective media, as the name says, selects for the growth of specific one kind of microorganism and it restricts the growth of others. So it has this mannitol salt agar, as the name says, it has salt in it. Sodium chloride is given at 7.5%, which is more than the usual sodium chloride that is found in the TSA media. So that high salt would restrict or would inhibit the growth of many microbes, and especially your skin microbes won't be restricted, won't be inhibited, but all the other microbes would be inhibited when you are trying to grow them on MSA plate. So salt becomes the selective part, and then you have mannitol sugar. That is the differential part of this media. Some bacteria can ferment glucose, some bacteria can only ferment lactose, some other bacterial cells can ferment glucose, lactose, and varieties of sugars. So depending on what kind of sugar you provide and whether the bacteria can break down, you can identify the bacteria. Okay, so this mannitol salt agar, as the name says, has mannitol sugar in it. If the bacteria is able to break down the mannitol sugar, you will see that the lactic acid is produced in the media and it will change the color from pink to yellow. If the plate changes from pink to yellow, it's an indication that the bacteria is able to break down mannitol. And this mannitol salt agar media has an indicator dye which is going to change the color of the media. When you see an acid forming, the color of the media would be changed from red to yellow. Okay, this is pink or red, whatever you want to call it. Okay, all right, so here you can see, so this is the original plate, MSA original plate, and look at how you can see the change in the color when you have bacteria that can ferment the mannitol sugar. Okay, so in the lab we have some bacterial plates done by our students, and look at the color change. So this MSA plate has been divided into 3 parts, and we added E. coli on one side, Staph aureus on the other side, and Staph epidermidis on this side. E. coli is not a bacteria that is found on your skin. typically it is in intestinal bacteria, gut bacteria. So that's the reason you can clearly see that there is no growth wherever the E. coli was swabbed. So how do you do this? You take the E. coli and use the sterile swabs. You pick the bacteria, you add on the MSA plate, you incubate these plates at 37 degrees for 24 to 48 hours and look for the growth of the bacteria. As you can clearly see, E. coli cannot grow on MSA plate because high salt will inhibit the growth of the bacteria. Here in this quadrant, you have Staphylococcus epidermidis. Staph, whenever you hear the word staph, it's a skin bacteria. Staph epidermidis can grow on MSA plate, but it does not have the ability to convert mannitol sugar, to break down mannitol sugar to lactic acid. Okay, that's the reason wherever you have Staph epidermidis, you can see that the color of the media is not changed from red to yellow. But if you see at this area where Staph aureus was added, you can clearly see that Staph aureus has the ability to ferment the mannitol sugar, and it can change the pH, and that's why you see the change in the color from red to yellow. Okay, so this is positive for fermentation and also positive for growth, whereas Staph epidermidis is only positive for growth but not positive for fermentation. So this would be your very first indication that the patient might have Staph aureus infection. But how do you confirm it? MSA test is not going to be a confirmatory test for Staph aureus infections outside. Okay, so the gold standard test for confirming the presence of Staph aureus bacteria is known as coagulase test. Okay, so let's talk about the coagulase test. Coagulase test. You have, these are the test tubes and they have rabbit plasma inside them. Staph aureus it's an opportunistic pathogenic bacteria. So it has the ability to convert the plasma into blood clots. Any blood plasma, it can, if it gains access into your blood cells, what Staph aureus can do is clot the blood. And what's the use of clotting the blood? The Staph aureus bacteria can hide inside the clots and escape your immune system. Okay, so this is the gold standard or the positive or the confirmatory test for identifying Staph aureus infections. You add Staph aureus to the rabbit plasma, and if the rabbit plasma coagulates or it clots or it becomes like a solid, then you can certainly tell that it is Staph aureus indeed. It's not something else, it's Staph aureus. All of the staph bacteria are divided into two groups: coagulase positive, coagulase negative. Okay, so the clotting is done because of the enzyme coagulase. Okay, so you see a clot, it's coagulase positive and it is Staph aureus. Let's take a quick look at the coagulase tubes or the rabbit plasma tubes. So this is rabbit plasma, which you can clearly see it's liquid. Okay, before we add bacteria, before we add Staph aureus. After we add Staph aureus and incubate this for 24 to 48 hours, you expect this. See, this is after we added Staph aureus. It's completely solidified. Staph aureus has coagulase enzyme and it clotted the rabbit plasma. Okay? All right. So, you learned about the MSA test and the coagulase test. MSA is again selective and differential. Coagulase test is a confirmatory test for identifying the Staphylococcus aureus against other staph species of bacteria. Okay? Then let's learn about another test which is known as blood agar test. Blood agar. The plate looks like this. It's red in color because it has sheep's red blood cells. The media has sheep's red blood cells. And you can imagine sheep's red blood cells, they are very, very rich in all the nutrients, so they don't select for any specific bacteria. It's not a selective media, it's only differential media. Depending on the bacteria's ability to break down the blood cells, we can classify the bacteria as alpha hemolytic or beta hemolytic or gamma hemolytic. So this is the blood agar plate after we add the bacteria and after we incubate it for 24 to 48 hours. On the right hand side, you can see you have Staph aureus bacteria which produced a zone of clearance. So all the blood cells are lysed here. That's gamma hemolytic. Sorry, that's beta hemolytic. Complete lysis. I know this is slightly confusing. Beta hemolytic is complete breaking down of the blood cells. Gamma hemolytic is there is no breakdown of the blood cells. Alpha hemolysis is partial breakdown of the blood cells. Okay? So in this figure, you have on the right hand side Staph aureus which can break down the blood cells. So this is beta hemolysis. On the left hand side you have Enterococcus faecilis, which is EF bacteria. It has no ability to break down the blood cells. So this is gamma hemolysis. As you can see, probably it's not very clear in the PowerPoint. I can show you guys the plate. The bacteria will grow, but it will not be able to break down the blood cells. Okay? All right, let's take a look at the result plate of the blood agar. So this was done by one of our students. On the right side you can see clearly if you hold and look. So this is an example of a very well done blood agar plate. On the left hand side you have Staph aureus bacteria that completely lysed the blood cells. On the right side you have the Enterococcus faecilis. The bacteria just grew but did not lyse the blood cells. Okay? So the left hand side what you are seeing is beta hemolysis. The right hand side is gamma hemolysis. And the next gram-positive diagnostic test we are looking at is the bile esculin test. So as you can see in the picture, bile esculin test is selective and differential. The bile salts, they inhibit the growth of many bacteria, and esculin is the differential component where some bacteria can convert esculin to esculite, and that esculite would turn the media black. Okay? So what is the positive? So in this picture in your PowerPoint, though they did the test in slants, in our microbiology lab, we do this in the petri dishes. This is bile esculin before the bacteria was added. Let's take a look at the result. So you can clearly see here, this bacteria, though it grew very well on the bile esculin plate, it did not convert the esculin to esculite. So you still will see the plate is kind of dark green. Whereas this bacteria, it grew very well and it also converted the plate to almost black. Okay? Another gram-positive diagnostic test that is commonly done in microbiology labs is the starch hydrolysis test. So, you're seeing the starch hydrolysis agar plate. It's very similar to your TSA plate. It's actually nothing but your TSA plate, but we add starch in it. Okay? And this makes it differential only plate. So this is not a selective plate. Any bacteria can grow in it because it's nothing but your TSA plate. Because it has starch, it becomes a differential plate. And how can you identify the presence of starch? So bacteria, if they have the enzyme amylase, they can break down the starch. So here, by adding starch, you are identifying the bacteria that has amylase enzyme. Okay? So if the bacteria has amylase, the starch that is present in the media would be all broken down. And how will you identify whether the starch is intact or is it degraded? Go back to your chemistry basics. What will happen if you add iodine to starch? You will get a dark purple color. Okay? So the first step is to grow the bacteria on the starch plate. So this student has two different bacteria on their starch agar plate. On one side, you have the Bacillus cereus. On the other side, you have the Staphylococcus epidermidis. You have two different bacteria on the starch agar plates. Both of them grew very well, but then whether a specific bacteria has amylase or not, you will only know when you add iodine onto these plates. When you add iodine, what do you expect? Let's take a quick look at the picture here. So this plate, there are 1, 2, 3, 4, 5. There are 5 different bacterial strains. Everything grew on the starch agar plates, and once they grew, what you need to do is add the grams iodine, the iodine from your gram staining. So once you add the iodine, if you see dark purple color, starch is still there. So when starch is still present, the bacteria has no amylase enzyme. But look at this specific strain. This is, I think this is Bacillus cereus. So what really happened here is you can see that there is a zone of clearance. So the starch is all broken down, and that's the reason you do not see a dark purple color around this bacteria. So this bacteria has amylase and there is no starch around. This is positive for amylase and also positive for starch hydrolysis. So all the starch was broken down by amylase enzyme. Okay? So this is starch hydrolysis test. Once again, this is a differential only test. Okay? And just to make it easy, I have a small table here. So this would tell you the kind of questions you might expect in quiz. Starch agar test. You are looking for the presence of amylase enzyme, and you are testing whether the starch is intact or not. And what are you looking? You are looking at the purple color. Okay? If you have amylase enzyme, if it is positive for amylase, obviously the starch will all be broken down. So I am writing negative here because the amylase will break down starch. And if there is no starch, there is no purple color when you add iodine. If there is no amylase enzyme, the starch will still be present and you will see a dark purple color when you add the iodine. So this table is important for your Canvas quiz. I will ask you questions based on this. Okay? So a specific bacteria, if it has amylase, what happens to the starch, and whether you get purple color or not, those kind of questions. Okay? Alright, so that's about your gram-positive diagnostic test. Last but not the least, there is one very easy test that can be done on the day itself. You don't need to wait for the bacteria to grow, and that test is known as catalase test. Okay? I'll show you guys the picture of the catalase test. So this is the last gram-positive diagnostic test we'll be learning today. You are testing whether the bacteria has catalase enzyme or not. Catalase enzyme is present in many of the aerobic bacteria. The aerobic bacteria, they should either have catalase or peroxidase or superoxide dismutase. Okay? So if the bacteria has catalase enzyme, the catalase would go and break down the toxic byproducts of oxygen metabolism. Okay? And one such toxic byproduct is hydrogen peroxide. So what you do is you take the petri dish like as it is shown here, and you add the hydrogen peroxide. If the plate bubbles, then the bacteria has catalase enzyme. Why do you think there are bubbles? Catalase enzyme would break down hydrogen peroxide to water and oxygen. So those bubbles are nothing but oxygen bubbles. Okay? It's very simple. You add the substrate, which is hydrogen peroxide, to the bacteria. If you see bubbles, the bacteria has catalase enzyme. If you don't see bubbles, the bacteria does not have the catalase enzyme. It's as simple as that. This is, as you can read, it's instant gratification. So this is one test that you can do just right on the spot. You don't need to wait for the bacteria to grow. Okay? Alright, so we are all done with the gram-positive diagnostic test. You learned about MSA, you learned about bile esculin, you learned about blood agar. coagulase test, catalase test, and starch agar test.